Thank you, Steve. And my wonderful collaborators, Dr. Steve Cox, just introduced some exciting findings related to this clinical trial on HEDS. And as a part of this clinical trial, today I'm going to share some preliminary data in using the personalized brain imaging techniques to explore how HEDS affect the human brain. So before we jump into the findings, I would like to give a quick overview of the brain imaging techniques that we used in this study. And can I ask how many of you have had MRI scan before? Oh, there's a lot. Yeah, and how many of you have had resting state functional MRI scan before? Much less. I would say my colleagues have the resting state functional MRI scan. So I would say most of you have had the structural MRI scan, which is commonly used in clinics to capture the brain dynamic structures. But for the resting state functional MRI, it's a little bit different. It captures the both signal changes, which reflect the brain activity changes. What is the both signal? Both represent the blood oxygen independent level independent. And this is also, complicated. And how to understand this easily? So when a brain region is more active, it will use more oxygen from the blood flow and leads to an increase of the both signal. And in the contrast, if neural activities goes down, the both signal will go down as well. So that's the basic theory. And now, if we extract two brain regions from the resting state functional MRI, and we will get two both signals, right? And then we can calculate the correlation between these two both signals to examine how similar these two signals look like. And we call it functional connectivity. If we got a higher correlation, we would see that these two brain regions may tend to change in sync over time. So that means at the same time point, if the brain activity goes up, and the other brain regions will also goes up. If they goes down, the other regions will also goes down. Then we call these two brain regions functionally connected with each other. So by using this functional connectivity method, Dr. Biswal and colleagues first identified the motor functional connectivity and reviewed the motor functional networks using the resting state fMRI data. So after a couple of years later, Dr. Yeo and a colleague using a similar method that calculate the functional connectivity across the whole brain, and then use a clustering method to group all the brain regions that had a similar functional connectivity pattern together and identified the 7 intrinsic functional networks in human brain, which we have the visual network, somatomotor network, dorsal attention, ventral attention network, limbic network, and also the frontal parietal control network, default network. So since then, these functional networks has been widely used as a brain atlas for clinical research. But if you look at this method, you will find some limitations, just as no two leaves are exactly alike. So the limitation is, for this group level functional networks, it can't capture the individual difference in the brain organization. So how to solve this? So our group developed an individualized parcellation method that allows us to reliably segment the human brain into different functional networks at the individual level. And as shown on the right panel, if you look at the functional connectivity within the same subject, but from different scans from different days, they look consistent. But between different subjects, these functional networks varies in shape and size. So after a couple of years, we have further optimized this method, and now we are able to segment the human brain into 92 functional brain regions at the individual level. And based on these brain regions, we can also calculate the functional connectivity across 92 ROIs. So this method provides us a powerful tool to investigate the abnormal brain circuit changes in brain disorders. So, of course, we apply this method to our clinical trial, EDS clinical trial. And for the brain imaging part, we collect the structured T1 images and also the 24 minutes resting state functional MRI, respectively at the baseline and also after the 1 month TMS treatment. So currently, there is limited understanding of how HEDS affect the human brain and which brain regions are associated with HEDS symptoms. So to answer these questions, we use this individualized parcellation to each participant's fMRI data at the baseline, and then we get these personalized brain regions as a seed and calculate the functional connectivity between these seeds. And as we know, dysautonomia symptoms are common symptoms in individuals with HEDS. And then in order to understand which brain regions is associated with dysautonomia symptoms, we perform the correlation analysis. And we calculate the correlation between the individualized functional connectivity and also the COMPASS score, which represent the dysautonomia severity. And interestingly, we found that functional connectivity of bilateral insular and also sensory motor cortex is significantly associated with dysautonomia symptoms. And then we average the functional connectivity within these regions and use it as imaging biomarkers. And we found that there is a strong positive correlation between these imaging biomarkers and also the dysautonomia symptoms. And that means higher functional connectivity in these regions may be linked to the more severe dysautonomia symptoms. So higher the worse. And then after 1 month TMS treatment, we found a significant reduction in dysautonomia symptoms in HEDS participants. And then we are trying to test whether the brain imaging biomarkers we found from the baseline can predict and track these changes in dysautonomia symptoms. Then we calculate the functional connectivity changes within the same brain regions and find it's positively associated with the dysautonomia symptom changes. So that means the more reduction in the functional connectivity after treatment, the better improvement of the dysautonomic function improved. So this means decreased functional connectivity and better. So what is the potential neuromodulation mechanism under this treatment? As we know, insular is plays key roles in the interoception and also the autonomic regulation. And the sensory motor cortex is associated with proprioception and also the somatic autonomic integration. So abnormal increased functional connectivity between these two regions may contribute to abnormal top-down autonomic control, and then further potentially increasing the dysautonomia severity. And findings from our other previous studies indicated that TMS can inhibit the brain activities in the sensory motor cortex. As you can see on the right corner, the blue regions represent the inhibited region, and it overlaps well with the sensory motor cortex. And when apply this TMS to HEDS participants, this may reduce the functional connectivity between the insular and the sensory motor cortex, and in turn, also reduce the dysautonomia severity. So all these findings suggest that TMS may be able to renormalize the insular and the sensory motor interactions and then improve the autonomic function in HEDS populations. So for the future directions, since we have already reviewed these brain regions that related to the dysautonomia symptoms, so we may be able to use different neuromodulation method to directly stimulate these brain regions. For example, TMS or TFOS, the ultrasound, we can maybe use these techniques to modulate the brain activations in these regions and then further improve the autonomic function in HEDS, and it can help the treatment for HEDS. And also, since we found these brain imaging biomarkers, we can use these biomarkers to track and predict the HEDS treatment. And it can help us better understanding when should we stimulate the brain, when should we treat the HEDS patient, and then when should we stop. And so combine this personalized brain imaging technique and also the brain stimulation techniques together, it will shed a new light for the treatment of HEDS. So lastly, I would like to thank our fantastic research team, and also all the brain imaging analysis was running through the MUSA computational core. So if you are interested in this novel brain imaging techniques, please feel free to contact me. And also, thanks to the Blue Sky Award, which is sponsoring our clinical trials. Thank you.